Morphological and anatomical study of the Pinus species. It is a supplied specimen. So far as the external morphology of Pinus is concerned, Pinus is a large perennial evergreen plant. Branches grow spirally, and thus the plant given the appearance of a conical or pyramidal structure. Sporophytic plant body is differentiated into root, stem, and acicular, that is needle-like leaves. A taproot system is developed there, and after the disappearance of this zone, the lateral roots are developed from the lower part of the stem. The stem is cylindrical and erect and remain covered with bark. Branching is monopodial. Two types of branches are present, that is long shoots and dwarf shoots. These are also known as branches of limited growth and branches of unlimited growth. The leaves are of two types, that is foliage and scaly. Scaly leaves are thin brown colored and scales-like and develop only on long as well as dwarf shoots. Foliage leaves are present at apex of the dwarf shoots only. These foliage leaves are large, needle-like, and vary in number from 1 to 5 in different species. Regarding the anatomy of the pine leaf and pine, that is the pine needle and stem is concerned, we have to go for the transverse section of the leaf and stem and observe under microscope. That is the anatomy TS of needle, that is the foliage leaf. It is circular in the outline in Pinus monophylla, that is semicircular in Pinus sylvestris and triangular in Pinus longifolia. Outermost layer is the epidermis. The epidermis is the outermost layer, which consists of thick walled cells. It is covered by a very strong, that is the outermost layer is the epidermis, which consists of thick walled cells and covered by a very strong cuticle. Many Stanken stomata are present on the epidermis also. Each stoma opens initially into a substomatal cavity and externally into a respiratory cavity or vestibule. Below the epidermis are present a few layers of thick walls, sclerenchymatous hypodermis. It is well developed in the ridges. The transverse section of the long shoot shows outermost single layer thick wall epidermis is heavily cutinized and followed by multi-layered cortex. The cortex is formed of parenchymatous cells. Cortex has a large number of resin canals. Vascular bundles are conjoint, collateral, and open. They form a ring around the pit. Regarding the Pinus needle anatomy, this is the leaf anatomy, that is leaflet. Epidermis is covered by a thick layer of cuticle. Then sunken stomata are present on the lower epidermis. The mesophyll of the leaf is, that is the mesophyll layer is the parenchymatous. Endodermis is present outside the pericycle layer, and the cells adjacent to xylem are called tracheidal cells. Regarding the stem anatomy of the Pinus stem, it is so much resemble with the dicot stem. In there 5 to 10 vascular bundles have been observed.